Hi, I'm Eric Wang,
And I'm Lily Tong.
and our research project was focused on investigating the effect on temozolomide on CPEB3 expression in glioblastoma cells. So glioblastoma multiforme is the most aggressive malignant primary brain tumor with poor prognosis, meaning that patients often have a short survival period. And glioblastoma, or GBN, accounts for 50% of all brain tumors, and on average, in the 40-year-old age group, one patient lives on average for 8 months.
So, the history of FDA approved treatments for glioblastoma is still very limited. Previously, there has only been chemo and radiation approved, and then in 1999, a new oral chemo drug called temozolomide, also known as TMZ, was FDA approved and is currently the most commonly used chemotherapy drug for GBM. And for our project, we will be focusing on TMZ to treat GB-derived cell lines. So what is TMZ? Also known as temozolomide, it is an alkylating agent that causes DNA methylation and the inability of DNA repair mechanism to fix damages, which will result in self-death or apoptosis. It is an oral drug commonly used for GBM patients and is a non-specific chemotherapy treatment in terms of cancer-related genes, which means that it doesn't target specific genes such as oncogenes and tumor suppressor genes.
One gene of interest in our project is the cytoplasmic polyadenylation element binding protein 3, or CBEB3, and this is a tumor suppressor gene that's often downregulated in various types of cancer, which includes GBM. And our previous studies have shown the presence of a CPEB3 ribozyme that is derived from the intron between exons 2 and 3 of the gene. And this ribozyme acts to self-cleave the gene, cleaving it in the form of pre-mRNA and preventing that from translating and synthesizing protein. While we do have plans to use more specific cancer treatment to potentially target this ribozyme, here we are only researching the effects of TMZ as treatment in vitro. TMZ is often used as the first line of treatment in GP patients, so we must first observe the effects of TMZ on CPEB3 expression before applying specific treatments to target the gene.
So our hypothesis is that we hypothesize that TMZ will decrease the gene expression of CPEB3. This is partly to show proof of concept of the chemotherapy drug on overall DNA expression inhibition. So methods. So U251 and LN229 are two cell lines derived from glioblastoma cancer. In the first round of cell culturing, we plated U251 and incubated for 24 hours. Then we treated one group with different concentrations of TMZ and another with DMSO as the control. DMSO was used as a control because TMZ was initially suspended in DMSO. The cells were then incubated for 48 hours and XTT assay was performed to determine cell proliferation and the optimal concentration of TMZ treatment. So in the second round of cell culturing, after figuring out the optimal concentration of TMZ to use, we repeated the same steps 1 and 2, except this time we plated both LN229 and U251, and we only treated with the optimal concentration of TMZ and again DMSO as the control. Then on day 3, we harvested the cells and split them into two groups for DNA and RNA extraction.
To continue the RT-qPCR process, we split our samples into two sets, which one set was extracted for DNA, for genomic DNA, and the other set was extracted for RNA and run through reverse transcription to obtain the complementary DNA. And both sets were then run through a qPCR, and we recorded the amplification of the CPEB3 gene, and this data was used along with the primer standard curve to determine relative gene expression. And we ran a gel to confirm that the gene amplified was the one that we wanted and did not amplify anything else. So our results from the XTT assay, we obtained the dose response curve for U251, and it shows that all concentrations of TMZ had significantly lower cell proliferation compared to the control, which suggests that TMZ does inhibit cell proliferation and causes cell death. And also from this curve, we determined that 150 micromolar was the best concentration to use moving forward. So we did use 150 micromolar TMZ, and when we obtained the CPEB3 expression, it almost reached significance of being lower in the TMZ group compared to the control. and we can generally say that it did decrease its expression. For the LN229 cell line, it did significantly lower the CPEB3 expression compared to the control.
So after the qPCR, we ran an agar shell to verify the correct qPCR products were amplified. And as you can see in the image, since there is only one distinct band, this verifies that our qPCR product was indeed amplifying the desired gene of interest, which was CPEB3. And here is a graph for the cell count of LN229. And as you can see in the graph, there's a significant decrease in the TMZ treated cells compared to the control group. And we also measured the cell doubling time. And in the TMZ treated cells, there is a longer doubling time compared to the control, which suggests that TMZ does inhibit cancer cell growth. And here's the graph for the cell count graph for U251. And as you can see, there is also a significant decrease in cell counts at days 2 after the treatment. And we also again measured the doubling time of the cells. And again, the TMZ treated cells had a longer doubling time, which suggests that TMZ does inhibit cancer cell growth.
So to conclude, we found that CPEB3 was downregulated in both cell lines when treated with TMZ, and that in the U251 cell line, concentrations of 600 micromolar TMZ or greater killed the majority of cells. And the overall cell count, lower cell count that we found in both cell lines is proof of concept that the chemotherapy drug did work as intended to kill the cells. So moving forward, we do plan on expanding our treatment regimen to include antisense oligos that would target the CPEB3 ribozyme specifically, and this would be treated along with TMZ to observe any synergistic effects.
So in the future, we also plan on culturing cells from patient-derived glioblastoma stem cells, in which the patients either had undergo chemotherapy or have not yet undergo chemotherapy. So with our current data, we can compare and align other treatments with TMZ to find the best care against glioblastoma cancer. And we also plan on moving our studies into in vivo mice trials in the future. So we would like to thank Dr. Claire Chen and Dr. Andre Luptak for providing us with the necessary resources to perform our experiments and acting as a mentor on this project. And here are the list of references we used for this presentation.
Thank you.
Thank you.